This molecule on the right is one of the most recognizable and important biological molecules. It is of course DNA, which is short for deoxyribonucleic acid. This is an important molecule because it's the molecule of heredity. It's inside the cell of every living organism and passes genetic information from one generation to the next. DNA is a large molecule which actually consists of smaller subunits, which are referred to as nucleotides. And nucleotides are made up of 3 different chemical units. One of these chemical units is a phosphate group, which actually forms the backbone of a strand of DNA. The second unit is a deoxyribose sugar, which is bonded to the phosphate group. And the deoxyribose sugar is a 5-carbon sugar which derives the name of deoxyribonucleic acid. And lastly, a nitrogenous base, which is bonded to the sugar. I'll circle a nucleotide in a strand of DNA here. And you can see the phosphate, the deoxyribose sugar, and the nitrogenous base. Now this nucleotide is then bonded to another nucleotide, and then that continues until you have a strand of DNA where you might have hundreds of thousands of nucleotides bonded together. Individual nucleotides are bonded together with what's referred to as a phosphodiester bond. Which holds the nucleotides together in an individual strand of DNA. DNA consists of 2 strands of nucleotides. So here is one strand here, and second strand here. And those 2 strands are bonded together in the middle by a hydrogen bond that forms between 2 nitrogenous bases. There are actually 4 different types of nitrogenous bases in DNA, which are adenine, represented by the letter A, thymine, represented by the letter T, guanine, represented by G, and cytosine, which is represented by C. And in the bonding between the 2 strands of nucleotides in DNA, adenine always pairs with thymine, and guanine always pairs with cytosine. This is referred to as the base pairing rule. What it means is that DNA is always going to have complementary strands. In other words, one strand always complements the other because of adenine always bonding with thymine and guanine always bonding with cytosine. When nucleotides are joined together into a chain like they are in DNA, opposite ends of the strand are designated numbers so that we know which end is which. One end of the strand is designated the number 5', and the other end of the strand is designated the number 3'. Now what you might notice is that the secondary strand of DNA on the opposite side, all of the bases are actually written upside down. All the letters are upside down. That's because in order for DNA strands to actually bond together, one of the strands of DNA has to run parallel but in the opposite direction to the first strand of DNA. So in this particular case, this would become the 5' end, and the other end would become 3'. So this is kind of like 2 trains running on a track in opposite directions. What this means is that 2 strands of DNA are complementary but anti-parallel. This is so the bases can actually face into the middle and bond with each other. This actually results in the two strands of DNA twisting around each other, like taking the top and bottom of a ladder and twisting it, where the rungs are the nitrogenous bases that are bonded together. And this forms its famous double helix shape. Another important nucleic acid is RNA, which is short for ribonucleic acid. And ribonucleic acid is made up of nucleotides just like DNA, except the sugar in RNA nucleotides is a ribose sugar rather than a deoxyribose sugar. So ribose is where ribonucleic acid gets its name from. These sugars are both 5-carbon sugars, and you can actually number those carbons in those sugars. So this is carbon number 1, 2, 3, 4, and 5. And what you'll notice that in the ribose sugar, on the second carbon is an OH group, which is bonded to that second carbon. Whereas in deoxyribose, the second carbon is bonded to a hydrogen group. So that's the difference between the ribose sugar and a deoxyribose sugar. Like DNA, RNA also has 4 nucleotide bases, except thymine is replaced by uracil in RNA, which means that adenine pairs with uracil in a complementary strand. Another difference between RNA and DNA is that RNA is generally a single strand, whereas DNA is, of course, a double strand. The function of these two molecules is related but quite different. So DNA has a few functions, but primarily DNA has a code in it which helps to code and create RNA through a process called transcription. Whereas RNA contains the code to create proteins through a process called translation. DNA also transfers information to daughter cells through the cell cycle, and we'll get into these processes in much more detail.